All right, so start with the test. How many people say microalgae are plants? True. Okay, we have some. How many people say microalgae are animals? How many say they're neither? Oh, wow. So they're actually kind of all of those things in my point of view. There are, plant-like characteristics and animal-like characteristics. As we heard, they're modal. They swim around. They run away from predators, not so much bivalves, but in fact, they're more primitive than either plants or animals, and there are evolutionary lineages from the green algae, the chlorophytes, to all land plants. So green algae are most like land plants, and then there are lineages like the dinoflagellates that are more closely aligned with animal evolution. So that's, if you thought they were one or the other, they're actually both in a lot of ways, but they're neither. Definition. So you're going to hear me and everybody else kind of use the terms microalgae and phytoplankton interchangeably, and those words etymological origins in different languages, but microalgae is the Latin definition. It's typological. It literally means little seaweeds. And when they were first observed, the algae were known. And then the things you couldn't see without a microscope were discovered, and they were called little seaweeds. So that's what that is. Phytoplankton in Greek is light wanderers, and that's more of a functional definition. That they use light, and they are not fixed in one place. They move physically by one reason or another. So here's an operational definition that I like for microalgae, and I'll point out that phytoplankton is specific to in the water, but microalgae in the broader sense is what I'll define here, and that is that they're single-celled or colonial photosynthetic aquatic microorganisms. That's a bunch of words, but I'm going to disrupt this definition and let you know what the significance of each of those descriptors would be. So they're single-celled. What this means is that when they reproduce by cell division called mitosis. And I'll find out now if I know which button to push for the light. Yeah, here we go. Okay, so one cell gets a little bit bigger, divides into two. These two cells divide into four, 4 into 8, 4 into 16. That's mitotic divisions. It's a way that the microbes reproduce themselves because they're single-celled. So the entire individual is one cell. The other way of reproduction is called meiosis, and that's where usually in multicellular organisms, but in fact there is recombination and meiotic divisions in some microalgae. But the fact that they're single-celled means that each individual is one cell, and a consequence of that is when you talk about population growth in microalgae, you're talking about there being more of them rather than bigger. So most things when they grow get bigger. With microalgae, you get more of them. Again, it's obvious to a lot of you, but it's part of how we define them. So they're photosynthetic. That means they're using this chemical reaction called photosynthesis to photosynthesis is really simple chemistry. It takes water and carbon dioxide, and there's actually a chemical in here that allows this reaction to occur. It produces sugar and oxygen. And the chemicals that allow this reaction to occur are photopigments, the most common of which is chlorophyll A. There's chlorophyll B, there's chlorophyll C, and there's all kinds of other pigments that may either help shield the alga from light or may assimilate and accept the oxygen that's produced by photosynthesis. But the color that someone mentioned they appreciate about microalgae is attributable to these pigments that are involved in the interaction between light and the organism through photosynthesis. So these pigments belong here. So that's the next part of the definition. They use light. And then they're aquatic, and I say that with a provision here. My colleague Pat Tester had a great statement about microalgae. Are they cosmopolitan or just everywhere? So microalgae can be found in the snow. There's red snow in the Arctic that is caused by microalgal growth. If you have a north-facing wall in your house, at least in middle latitudes, you probably have algae growing on your house. My PhD advisor, Frank Trainer, had a shoebox full of dirt from the ground at the University of Connecticut, and every year for 40-something years, he had his students take a spoonful of that dirt, put it in algal culture media, and there were blooms of algae. So the microalgae are not just aquatic, really, like phytoplankton, we call, those are the aquatic ones, but microalgae can be found everywhere. This is interesting from an ecological standpoint, but it's also a possible source of problems when you wonder where contaminants come from in your algal cultures. And the fact is the pressure of contaminants comes from everywhere. There are two basic cell designs in microalgae. The one on the right here is the very simplest and the most primitive. We call it, the proper term is cyanobacteria, but we call them blue-green algae. And how many people have had blue-green algae grow up in their algal cultures instead of what they're trying to grow? Yeah, I mean, it's because they're very aggressive and tend to be not very good nutritionally, which we'll say something about later. But this is a very simple design. As you can see, the DNA and ribosomes and the photosynthetic pigments are all in one bag. So it's a bacterial cell design. The other model of microalgae is more complicated, and most of the ones that we deal with in hatcheries are eukaryotes. That means they have a nucleus where most of the DNA is contained, and then they have the photosynthetic and heterotrophic functions in separate organelles. And so you have simple cyanobacteria that are prokaryotes, and most of the algae we're interested in from shellfish standpoint being eukaryotic or having a true nucleus and having a more complex internal structure and more complex internal function as well. So the next thing I'm going to do is show you a lot of photos of the different kinds of microalgae. The most common classes, there are now, I think, 49 taxonomic classes of microalgae, but somewhere around 35 of them each have only one individual. And how we got to that stage in understanding the evolution of these is something I'll mention at the end of the tour through the zoo here, the microalgal zoo. This is a spring bloom photo from Milford Harbor, and I can look at this and see some little nichia thing here. There's a lot of skeletonema chains. There's a Chaetoceros chain. There's probably, in most natural assemblages, there'll be maybe 3 dominant species and about 20 taxa that are present in small percentages. And kind of what the consensus of the phytoplankton ecology community is 3 to 7 days is turnover time of who's dominant. So it's very dynamic. There's tens of thousands of species of algae described, and probably 5 to 10 times as many that exist. So there's a lot of diversity. So I'll try to again cover the main classes in a number of slides. So here's what cyanobacteria look like, or the blue-green algae. And the first thing you notice from looking at representative photomicrographs here is that not many of them are present as single cells. These are only about 2 by 1 micrometer in size each cell, but they tend to make colonies, and they tend to be linear colonies or tufts. So the effective size in the environment could be a lot bigger than 1 or 2 micrometers. So they could be easily filtered by shellfish because of their colonial nature. Chlorophyll green algae, I mentioned these before. They're the evolutionary origin of all land plants. Bob Guillard used to say that a tree is just chlorella on a stick. In terms of cell physiology and so on, that's pretty much true. And these we tend to encounter. We don't use many of these on purpose, but when we encounter them as contaminants, a lot of us have called them LRGTs, which is an acronym for little round green things. They're really hard to identify. These are only about 2 to 3 micrometers. That one is, smaller individuals from this Chlorococcus population are only a micrometer or so in size. And you see, they don't, this one is not colonial, but there are some colonial ones. There are 2 other kinds of several other green algal taxa that are from completely different evolutionary lines. And here's a photo that I used forever, but I really like it. So this one is Denaliella, which is a chlorophyte. It's in that green algal group. This one is Tetraselmis, and I know that because when the cells divide, the 2 daughter cells stay in the mother cell wall for a little while, so you get these doublets you can see in culture. Denaliella is a completely inefficient or incomplete diet for shellfish, and some Tetraselmis strains are the most complete diets we have. And this one is in the green algal group. This one is in a group called the Prasinophyites. And these were brown algae that lost the yellow pigment that makes them brown later on in evolutionary history. But if you look in the microscope, it's pretty hard to, if you didn't see dividing cells, it would be like pretty hard to identify them from each other. So morphology is not a good indicator of nutritional value or even of the evolutionary placement of an alga. Okay, another group that we're really interested in is the Primnesiophytes. And these are flagellated brown algae. They're basically, I'll talk next about diatoms. They're diatoms without shells and with flagella in terms of their cell physiology, in terms of their pigmentation and so on. And a really interesting group. This includes the T-iso that a lot of us use, or C-iso, and Pavlova is usually lumped into this group. It also includes Primnesium that kills sea life in Gulf states, especially in Texas. So again, you can't take a group and say, well, these are all good, because there's usually some bad players in every big group of algae. But it's kind of ironic in a way that the whole class was named after the toxic one, Primnesium, Primnesiophytes. Diatoms come in 2 basic. These, I say, are brown algae with glass shells. These are the centric ones, and the ones we tend to use. They tend to be planktonic, so they've got different ways of keeping themselves up in the water rather than sinking and growing on the sediment surface. This one's Chaetoceros. This one is 3H Thalassiosira. Melisira's down here. Here's a Thalassiothrix, I think. So these are, this is a group that we're really interested in from standpoint of shellfish feeding, just like the Primnesiophytes. So this whole clade of brownish, yellowish colored algae tend to be good foods for shellfish. And, we'll say a lot about that later, but the other basic model of diatoms is the pennate ones. The centric ones have radial symmetry, so at some orientation you could draw a line through them anywhere and have 2 sides be the same. Bilateral symmetry is more like a side and a left side. And these include Nitzschia, Grammatophora, that's Thalassiothrix, I think. I always mix those up. But anyway, these tend to be benthic. They tend to grow on the sediment surface, and I imagine they might be present in waters here. I've read some papers where this could be quite a big source of food for shellfish in shallow places where the water is clear. The water isn't very clear right here, but some of you may have places where you're growing clams. And these get mixed up by tide or wind currents and wind up in the plankton for a short time before they sink again and get eaten. We tend to not use them for feeding shellfish. There was a long time ago, people used a member of this group called Phaeodactylum, or it was called Nichia clusterium also. But we actually discovered, or other researchers discovered, that Phaeodactylum can be a little toxic. So it doesn't get used very much anymore, and I don't encourage its use, but some people still actually grow that. But the other kind of diatom, they're harder to keep in suspension, so you need more mixing to keep them up. Another group, the cryptophytes, are really complicated morphologically. If you look at some of these photomicrographs and this diagram here, there's all kinds of internal structure in cryptophytes, and I'll tell you in a minute how that got to be the case. But these tend to be red in color, and people, when they see the natural bloom of them, freak out and say, "Oh my God, it's a red tide," and so on. But most red water in where I live in Connecticut tends to be cryptophytes, and I don't know how common they are down here. Do you ever see red water that's not dinoflagellates? Anybody? Okay. Well, these are notoriously difficult to manage. They're unforgiving. We grow them for some applications that have not much to do with shellfish feeding. And I say they're unforgiving because the minute they run out of nutrients, they die. And so you have to, keep on them. You have to keep harvesting. You have to keep transferring them. They're a pain to keep. We have a culture collection of 230 something strains, and the cryptophytes are the hardest ones to keep because you let them go a day too long and they're finished. And so most people, they may, some of them are pretty good nutritionally, but most, practical hatcheries or people growing them for practical reasons don't try these and give up on them pretty quickly because they're hard to manage. And then there's a eustigmatophytes, which are also in the brown algal clay, like the diatoms and the promnesiophytes, but they tend to be small and they are green in color because, like the prasinophytes, they've lost the brown pigment. And these are also among the LRGTs. They're pretty hard to tell morphologically from little chlorophytes. These are frustrating because their biochemistry is really good for nutrition bivalves, but they're usually quite indigestible. And I don't know, how many people use Nanochloris or Nanochloropsis, something? Yeah. So there's a possibility that clams can actually digest these better than other bivalves, and it's not something that's been tested critically, but we know from for oysters and bay scallops that they just simply don't digest them. Clams may, again, no. Yeah. That's right, exactly right. That. Yeah. So right, larvae, I'm pretty sure don't. Right, but post-set, maybe the style is a bit tougher in clams than oysters or scallops. But anyway, these grow really well. Their biochemistry is great. If someone can discover a way to make them digestible, they'd be wonderful. And I actually had a grad student try some enzymes to see if we could get through the cell wall, and we didn't happen upon something that worked. And then the famous dinoflagellates, which this is the true red tides. In New England, we have this this character here, Alexandrium catenella, which has changed its name recently from fundiense, which used to be called tamarinse. And then you guys in Florida, not right here, but how many of you encounter the Carina blooms? Yeah, I mean these are horrible in a way, but they're different evolutionary strategies that organisms develop to survive. Most of the microalgae survive by growing faster than they're eaten, and dinoflagellates have evolved with slow growth rates, but a way to inhibit feeding by bivalves and other predators, and zooplankton especially. And there's some great papers that have come out in the last decade that have shown that most of the microalgal toxins that bother people also bother copepods and other crustacean grazers. Some of them bother bivalves, as work that we did in our lab, and thereby allowing a slower growing population to persist if it's just eaten more slowly. As a consequence, they're not good choices for feeding shellfish on purpose, generally speaking, which is too bad because their biochemistry is very good. A lot of them would be good foods if they weren't toxic. And another very recent finding with dinoflagellates is we obsess about the neurotoxins that affect human seafood consumers, but a lot of toxic compounds that don't bother us but other marine life. Including other algae. So that's how they bloom. They exclude their competitors and kill their consumers. So it's a good strategy for ecology, and they've been around for a long time. But again, they're more of a pest for us than a resource. Okay.
We have this data set on the brevetoxin mollusk. What about microcystins from our photosynthetic bacteria or cyanobacteria? Microcystins.
Yeah, I mean, I don't know as much about those because there haven't been as many practical problems with them. But my understanding is that with freshwater research with freshwater crustaceans, that they do inhibit or kill, and it's maybe even not requiring feeding, because microcystins tend to be released in the environment. Okay. Another one, actually, I have a question about. Do you have Prorocentrum minimum this far? Oh, okay. Okay. Yeah, because my lab did a lot of work, and it was really frustrating because sometimes it's toxic and sometimes it's not. We finally figured out what induces toxicity, and it is actually nutrient deficiency leading to loss of photosynthetic function, and they switch from being photosynthetic to being heterotrophic and eating bacteria. And the enzymes that they use to digest the bacteria that they eat are toxic to shellfish. Talk about complicated story. The dinos are the most complicated microalgae on the planet. And anyway, that's some factoids for you on dinos. So all this diversity comes from hypothesized, let's say theorized. Theories are better than hypotheses, right? So we're pretty sure that the diversity in microalgae comes from an evolutionary process that started with something called endosymbiosis. And I showed you that slide a while ago of a prokaryote being a very simple design and a eukaryote being more complex with a bunch of stuff inside. And how that's thought to have happened is back in evolutionary history, some heterotrophic thing that was eating bacteria, including cyanobacteria, discovered that the function of these bacteria inside of the animal type cell that was eating them, that those functions were good. They kept working. The cyanobacteria kept producing sugar that was feeding the heterotrophic bacteria and producing proteins and functional enzymes and so on. And so, there's actually still present in the water organisms that do this temporarily, that will eat algae and keep them alive inside and keep them working, keep them producing sugar by photosynthesis and not digest them entirely. So it's thought that these consortium kind of organisms that have now persisted in this state, that they keep them forever, are that's the source of eukaryotic cells. And all the cells in our body are eukaryotic. In terms of evolution, you can when the multicellular organism, bacteria is single-celled. My contention is the bacteria be here a long time after we're gone, but so who's successful? We'll find out. Well, we won't find out, but it'll become apparent. So anyway, you know, this endosymbiosis theory is pretty much how scientists who study this think it happened. And here's like one of my favorite diagrams in the world. This shows the various symbiotic, endosymbiotic consortia that account for the diversity of microalgae. So you had primary endosymbiosis of cyanobacteria, and in 2 different clades. These are all the brown ones. I kept mentioning the brown algae as being, you know, a group that is good for us and so on. All of the dinoflagellates, the cryptophytes, the eustigmatophytes, the diatoms, the promnesiophytes are over here. The green algae and euglenas and, you know, things on this side are a separate whole direction of evolution. And interestingly, on this side, on the green side, the fatty acids that are dominant in membranes and in, well, mainly in membranes, tend to be omega-6. The unsaturated fatty acids have the first double bond 6 carbons in. It's just, you know, I'm not giving you much context for this, but, you know, you may have actually read in the news about human nutrition and omega-3 and omega-6 fatty acids. So the omega-6s are all on this side. The omega-3s are all on this side. This side tends to feed the oceans. And so the nutritional benefits to people of omega-3 fatty acids, all those fatty acids actually come from the bottom of the food chain, the beginning of the food chain, and brownish microalgae. These compounds you get for animals like shellfish that feed on microalgae. So the biochemical capacity to manufacture these compounds has been lost or is never developed in multicellular organisms. So we need this from our food, including humans. We need omega-3s for good membrane function, and so we need to eat seafood actually to be healthy, to get these compounds. And there's some, you know, famous percentage of the fats in our brain, which is basically a blob of fat up on top of inside our heads. That, you know, the omega-3s that are dominant in that organ of ours have to come from brownish microalgae on this side of evolution. But, you know, that's, you know, like expanding the thought out quite a bit. But I mentioned the dinoflagellates are especially complicated because there was a third endosymbiosis down here where some brown alga or a green alga is incorporated in a dinoflagellates, and so the dinoflagellates is the most complicated evolution, most complicated physiology and morphology. But another like major consequence of this is that the taxonomy of microalgae is chaotic. And it's becoming more and more so as molecular biology tools are increasingly applied. And you know, every time somebody goes prospecting for DNA sequences in the sea, you find 3 or 4 that are recognized from cultures or from, you know, from species that are described, and several thousands that have never been described. So, and then every other week, an organism well known like Alexandrium tamarense gets redefined, renamed as Alexandrium fundiense, and the week after that it's renamed as Alexandrium catenella. How many grow T-iso? Do you know what that's called? Huh? Yeah, you know the clone is T-iso. But what do you call it if somebody says, what genus and species is that? Isochrysis? Okay. Okay. So that was changed about 5 years ago. And actually, you know, this was a smart thing because it's not Isochrysis galbana. There's a good species called Isochrysis galbana. We have 3 strains in our collection. And biochemically, they're nothing like T-iso. And even morphologically, they're not very much like T-iso. So finally, a group in Roscoff wrote a good species description. It's now called Tysochrysis lutea. And again, that's a good species name. But in a way, it doesn't matter to us as people interested in feeding shellfish, because it will always be T-iso. And that's like a really important point that I want to emphasize here, that as you receive microalgal starter cultures from us, from UTEX, from your neighbor down the next bay, keep the strain designation. If they call it T-iso, keep calling it T-iso, because those strain designations don't change, even if the scientific name changes every 3 weeks, which sometimes seems to be. And this is a big point actually of frustration for me, because I'll get a call or an email from a hatchery that says, well, we lost our Tet strain, and we want a replacement. And it's like, well, I don't have anything in my collection called Tet. Oh, that thing you sent us 5 years ago, we started just calling it Tet. So how do you expect me to know what I sent you 5 years ago if you changed what you call it? So I'm begging you, keep the strain designations, and never mind. I mean, someone will correct you at some point whether the name has changed, genus and species. The strain designation is good forever, so please keep that. Yeah.
Make T-iso non-native or exotic to Florida waters. This is from a very important regulatory perspective that our hatcheries are now facing.
Yep. So you can't prove that it's not native. Can you prove that it's native? Well, if you looked hard enough, if you did, genetic prospecting in Florida waters, would you find that? My contention, this is how I answer that, is like, yeah, probably you would. Probably you would, but it's not been recognized. But, let's also, understand that only 1% of what's endemic here has been recognized, described, and named. Is it a risk? I mean, so there's two questions, right? The first one is it non-native? Let's say it is. What the heck? Okay. Now, is that a risk? What's the risk of that? So you look at the organism, it thrives in culture conditions of unbelievably high nutrients without contaminants. If you get a contaminant in a T-iso culture, your T-iso is gone in an instant. It is the most delicious thing. There's got no toxins. It's got, all good biochemistry. So if it gets loose and, takes over an ecosystem, the consequence of that is the ecosystem is better fed. So, is it a risk that we want to prevent? The worst case outcome is actually improvement in nutrition of the marine community. Is that a risk we're willing to take? And that's basically the only way you can.
Can we quote you on that?
Yeah. I mean, I've been asked that so many times, but that's okay. Yeah. That kind of discussion is good with beer. All right. Well, so this is the introduction to the algae, and lunch is not here yet. We could do one of two things. We can, take 5 minutes to chill, or I can start another section on function of microalgae. And what do you want to do?
Are there any questions right now?
Yeah.
So my question is, as you were speaking about names, the Tysochrysis lutea, you have T-iso and C-iso. So what is the difference?
I know that. I know the answer to that. So but I still need help to nail it down for sure. So C-iso was isolated in a lagoon on Providenciales in the Turks and Caicos Islands, right at the outfall of a conch hatchery that Megan Davis was principal in. And, you know, was called for Davis. It got put into culture collection. They regrew it there. Great, you know, it's good food for conch larvae. It got a bunch of collection, got sent to us. We cleaned the bacteria out of it, put it into Bigelow, and, you know, it's been propagated since then. You know, is it different than T-iso? I think it's the same thing, re-isolated from the babe and the outfall. But to be sure of that, I need the group in Roscoff actually to run run genetics, you know, and see how close they are. But there's a complicated story there, and that is, you know, T-iso has been in culture since the late 70s, because we got it in Milford from the guy who isolated it in Tahiti, separately from the strains that got sent to other people from Tahiti. And, you know, we can, to some extent, trace like who we from Milford's T-iso got sent to these collections and they propagated there. So you actually can trace the provenance of T-isos back pretty well, because there are pretty good records in our lab of who we sent strains to. How have they changed in the tens of millions of generations since it's been isolated and grown in Milford versus grown in Maine or grown in UTEX, you know, in Texas? And that's like an enormous, great question for a scientist. You know, how do strains change over time in culture? Do they? Right? It shouldn't. But what, you know, there's something called strain drift that some bacteriologists have put effort in. So I said, so I know the history of C-iso. I know where it came from, and I got it directly from the Caicos rather than, you know, through some other source that maybe it was mixed up or something. Practically speaking, you know, again, I think it's the same thing. It may be changed in Turks and Caicos in some way, and when it was re-isolated, had changed a little bit. But in terms of biochemistry, growth characteristics in our laboratory, it's the same. Those two are the same. And different people, including us, have better success with one or the other in large cultures. And what I tell people, you know, is try them both. The one that grows best for you, just go with it, because your shellfish larvae are not going to care which one, which one it is, because giving them, you know, biochemical constituents.
Great, thank you.
Okay. Anybody else? So what do we stop or do we keep going? Okay. All right. Oh.
Dr. Clay, who ran UTEC for quite some time, 20 years ago,
was quick to correct me in that there was no such animal as an exenic culture, and had speculated into a potential discrepancy from bacteria from T-iso as opposed to Manila C. Would there be anything to that as far as any other, if we're assuming that there is no such thing as an exenic culture?
So that word azenic is one that I hate also. And it means, you know what the word xenophobia? Fear of foreigners, right? So azenic means without foreigners. So it means there's only one organism in that culture. And, you know, I'm happy to use unialgal bacteria free, but unless I have done a very thorough analysis of viruses that are considered living, I don't think I can say azenic. Different reason for not liking that word. It's what you can confirm. I mean, you can confirm that there's a single algal species there by morphology, by genetics. You know, genetics is the right way to do it. You know, there's one eukaryotic sequence in there, good enough. You know, and then you can confirm bacterial contamination a number of ways, with flow cytometry, with microscopy, and again, the best way is with genetics. There's zero prokaryotic you know, sequences in there. But viruses are harder to be sure about, and none of us have done that. There's nobody who's done a thorough check for viral DNA in microalgal cultures. So I don't like the word azenic. I'll use unialgal bacteria free every time. Okay, there's actually a nice short chunk that we can do while that happens. So in the ecosystem, it was a little bit about functionality of microalgae. In the ecosystem, I like to call microalgae or phytoplankton transducers. Anybody know what a transducer is? Can anyone give me an example of a transducer that we encounter every day? I have a here in music, so I always go to microphone. It changes sound energy to electrical energy. The sound travels in a wire, and you have to plug it in, right? And then the loudspeaker. Energy goes as electricity into the speaker, and sound energy comes out. So a transducer changes the form of energy. So microalgae are transducers because they take the sunlight energy, and we talked about photosynthesis already, and they turn it into chemical energy. They turn it into a compound, sugar, glucose, that's got high potential energy. When it is oxidized, it releases that energy. So they're transducers, and they're actually pretty much the only organisms that do this, other than the macroalgae, and they participate in interactions with the chemical and biological ecology, and they're an essential part, because without the phytoplankton, this energy wouldn't find its way to all these other components of the ecosystem. So, you know, think of them as transducers. They're energy modulators or modifiers. And something I have a great big green block in here, and I didn't mention. Photosynthesis is a very simple equation that I already showed you. It just makes sugar and oxygen. For an entire cell to be produced and to be functional, you also need a bunch of other elements, nitrogen, phosphorus, the main ones, and then the carbon from carbon dioxide in the atmosphere. So there are also transducers of materials, dissolved nutrients to more complicated chemical compounds like proteins and starches and fats. This is another way to look at photosynthesis, which I like because it introduces an idea about stoichiometry, which I'll say a little more about. There's quite a lot more about it actually later. There's sunlight, there's water, there's carbon dioxide, and there's certain amounts of water and carbon dioxide that combine to make sugar and diatomic oxygen. So there's this thing called stoichiometry that I'll cover a little bit later. I'd like to show pictures of how photosynthesis works, just so you kind of get an idea of the challenges that the organisms are facing to move things around. So this is the simplest kind of photosynthesis that you find in most cyanobacteria. It's called C3 or 3-carbon photosynthesis. You have carbon dioxide that has to diffuse into the cell, diffuse into the chloroplast if it's a eukaryote, and this enzyme called rubisco changes the carbon dioxide into a form that can be assimilated by photosynthesis. So there's one enzyme or a class of enzymes called rubisco, and thereby allowing the production of all the other cell components. And if this is the only form of photosynthesis, it would be like very inefficient, and we would have a crisis every time the availability of carbon dioxide in the environment changed. This still operates in all microalgae, but it's not the dominant form. Here's the dominant form, and it involves another class of enzymes called carbonic anhydrases. And what carbonic anhydrases do, they do it inside the cell and they do it outside the cell, is to modify carbon dioxide to bicarbonate. Bicarbonate is transported by carbonic anhydrases into the cell and then changed back into carbon dioxide at the site of the chloroplast. So what carbonic anhydrases allow is for bicarbonate in the environment to be accessed by photosynthesis. It's like critically important evolutionary step was the evolution of carbonic anhydrases. And this is especially important if you look at something that scientists are obsessing about in marine and aquatic ecosystems. That's ocean acidification, or what's the carbonate chemistry like in the ocean. So if you dissolve carbon dioxide in water, if the water is very acid, it stays as carbon dioxide. As the pH increases, and here's neutrality, carbon dioxide dissociates to bicarbonate. And if you increase the pH much above 9, you see the bicarbonate dissociates into dissolved carbon dioxide. And here's about the pH of the ocean is somewhere around 8. And what you can see is in the pH of the ocean, the carbohydrate that is accessed by rubisco is essentially 0. So algae would not be able to function in the ocean today without carbonic anhydrase enzymes changing that, allowing the cells to access bicarbonate. And another interesting, I'm a contrarian about a lot of stuff in science. So here's an example of that. Here's the threat of ocean acidification represented in the gray bar. So it said that the pH of the ocean may decline, from above 8 to almost 7, and this is going to be catastrophic. That all life in the oceans is at risk, and so on. And you can see that change in pH does not change at all the availability of bicarbonate to microalgae. So humanity may be toast, and bivalves may be finished. I don't think so, but the microalgae would thrive in a neutral ocean as opposed to a slightly basic one. So please don't say no, it denies climate change. I'm just saying, and this is actually important also in a culture. And tomorrow, when I talk about chemical ecology, the presentation my colleague Shannon Nisik put together, there's a lot more about this. But a consequence of this is you need to control your pH. And actually, if the pH declines in your culture, down in the range of 7, it's actually not a problem at all in terms of carbon availability. If you allow the pH in your culture to go up above 9, bicarbonate availability tends to go down, and there's no way that dissolved carbon dioxide is accessed by photosynthesis. So actually, when I talk about people controlling pH, as algae take bicarbonate and carbon dioxide out of the water, they tend to drive the pH to the basic side. If you let that happen too much, you're going to cause a number of chemical problems, one of them being the availability of bicarbonate for photosynthesis. So culture acidification, not a big deal. Culture basification, something you need to be concerned about. And if you own, I usually say, what does the hatchery need? You need a pH meter before you need a microscope, honestly, because you're finished if the pH gets too high in your cultures. So here's some research from our lab about the effect of changing pH to the acidic range of culture acidification, effects on phytoplankton biochemical composition. And this was a hard paper to publish because they're all negative results. This is a C to N and fatty acid composition of many or several strains or several species of algae. And you can see as you get more and more acidic in the culture, you basically don't really change the biochemistry of them very much because they're happily in their pocket, their bicarbonate pocket. So that's like confirming what's kind of the obvious, that there shouldn't be a change. And then here's my little movie that I stole from the internet on the physical chemistry of photosynthesis. So this is inside a chloroplast, and that is the thylakoid membrane. that has chlorophyll and other compounds. And so photons enter that, kick off electrons. The electrons produce NADPH and ATP. And we'll get there after we get the stoichiometry up where we need it to be. Those 4 ATP and 1 ADPH enter the Calvin cycle, which you remember from freshman biology if you took freshman biology. And the 4 ATPs then phosphorylate the NADPH to NADP. And that cycle is what produces sugar. So again, it's just like a little cartoon, but there's a physical machine inside a chloroplast that assimilates the, I'm going to stop it by going forward. There's a machine in the chloroplast that assimilates the energy, the photons from the sun, in a way that is described and known, and in a stoichiometry, 4 ATPs, 1 ADP, and ADPH. Okay, so we know how much energy is needed to produce every little pump of energy that makes sugar in the Calvin cycle. So that's something you can calculate. You know, in a hatchery, you're not going to calculate that number, but if you recognize that if you're putting insufficient energy in, you know, relative to your expectations, that you're not going to get the rate of that machine running is not going to be the rate you want. So, you know, light limitation is a common thing that people run into for a bunch of different reasons that I'll talk a little more about later. So here's a stoichiometry of that, you know, how that machine runs. And I found this recently, and I think it's really interesting. So this is one way of drawing, a chemist drawing chlorophyll molecule. And you see there's a magnesium in the center of this, you know, lipidic ring structure. This is hemoglobin from human blood. It's the same compound with iron at the center instead. You know, and you know, so handling energy, handling oxygen, it was invented once by bacteria and modified in evolution. And this is like a huge lesson, you know, from a science standpoint, you know, that has informed like most of the research I've done in the last 20 years, that biochemistry is conserved in evolution from the simplest organisms. Every biological machine, every enzyme was invented by bacteria and assimilated, stolen, borrowed by other life. So, you know, and the microalgae are closer to bacteria in their evolution than we are, for example. So, you know, a lot of things that may puzzle you about the function of microalgae can actually be figured out if you understand something about bacteria and if you know the basic biochemistry of life. So it's a good, if those of you who have that knowledge, don't be afraid to apply it. Don't say, well, that happened. I know that happens in human cells, but does it happen in algal cells? The answer is probably. Okay, this is an ugly figure, and it's an Odom wiring diagram, but it shows the consortium nature of a microalgal cell. The cell is the bullet-shaped thing. Here's the chloroplast that's, or which is the chloroplast? Yeah, no, here's the chloroplast pumping out sugar, and here's a mitochondrion burning that sugar and turning it into the energy. There's a lot of recycling of oxygen and materials inside the cell and within the environment. And so again, the bacteria are fully functional inside the algal cell as they are outside in transforming the important elements of life: carbon, oxygen, hydrogen, and then the nitrogen and phosphorus that are the next most important components of living tissue. So this is almost unreadable in here, but oh well, that's the way it goes. So, you know, I mentioned stoichiometry, and here's where I develop it a little bit more. The stoichiometry of photosynthesis is here that you can't read. And then there's this thing called the Redfield ratio. Has anybody heard about the Redfield ratio? Redfield ratio is is a measurement. It's you know, not something first recognized theoretically, but there's a ratio of carbon to hydrogen to oxygen to nitrogen in living matter in the sea. Was where it was first determined. And you know, that's 106 to 16 to 1 for carbon, nitrogen, and phosphorus. And that ratio has been extended to this other thing that you can barely read to include a bunch of other elements. And there are actually, there's some 40 elements that are essential to every living organism. And these tend to be present in a very set ratio. And some of the important ones, you know, as we already looked, like magnesium is in the middle of chlorophyll. There it is one of the, you know, the important chemical components. Potassium is in there. Calcium is huge in cell physiology. So the basic message of the unreadable slide is microalgae not only need to get carbon, which I described, you know, at length, coming with, you know, a series of enzymes, but they have to access all these other elements from seawater, and they concentrate them by many thousands of times. So, you know, how does that happen? Well, there's 3 ways, and again, this is an unreadable chart, but the main elements of carbon and nitrogen and phosphorus are acquired, you know, first by passive diffusion, and then sodium, chloride, you know, some of the really big potassium, the really big components of seawater, plenty of it gets in by passive diffusion. When they have to work a little bit harder, which they do for carbon, but for a lot of other metals, especially divalent cations, there are enzymes that are induced by a low quantity outside the cell. So to acquire calcium, for example, if the calcium is very low in the environment, enzymes inside the cell will be upregulated that are especially sticky for calcium ions and will grab all the calcium ions that come to the surface. And these tend to be on the surface of the cell. And then there are the really hard to get things, and iron is one of these. Cobalt is another one, where extracellular enzymes that are produced all the time or are induced by starvation inside the cell. So chemicals that are released into the environment, the ones that access iron are called siderophores. They change the oxidation state of iron in the environment to a state that is more easily assimilated, and the siderophore iron complex is then assimilated into the cell. So there are, you know, complicated enzyme systems to allow algae to acquire the elements from the environment that they need in, you know, hundreds to thousands of times the concentration inside the cell they need compared to what's outside. So, you know, that's an aspect of microalgal physiology that we don't really pay attention to, but we really need to, because we add nitrogen and, you know, nitrate and phosphate, we add silicate for diatoms, and we make sure that the pH is in a good range for getting carbon, but we also add this thing called a metal mix. Yeah?
I know that Redfield came up with the ratio like that.
Yep.
Has there been discovery on like variability of the ratio, or maybe the extended ratio due to strains or whatever? It's been almost 100 years.
Yeah. So the question for those on the thing is, you know, has there been updates to the Redfield ratio? And I think so. And I can't say that I follow that very well. Something really interesting that we figured out is nitrogen to phosphorus ratios in the successful algal culture media, including F/2 and my boss Euclid's E medium, and you know, the bold medium and so on, usually 25 to 1 N to P, right? Not 16 to 1. And you know, the interpretation there is that phosphate is more efficiently recycled. The luxury consumption of nitrogen gets locked up in compounds that are not easily recovered by the cell. In other words, whereas the phosphorus, the recycling of that is better. So you know, that's the only thing I'm aware of. But the other elements, I mean, I know there's a lot of oceanographic work in different parts of the ocean. I really haven't followed it well enough. From an algal culture standpoint, we add metal mix. You know, we add metals to a hugely higher amount than the cells require. And then we put a chelator, an artificial siderophore called EDTA, in that mix. And that modifies the oxidation state of the metals and allows the cells to get them. But, you know, we still, you know, have this problem of how do algae get at this stuff that's outside of them, right? So a little bit on boundary layer physics, which is really fascinating. And again, I don't expect everybody to, like, you know, apply this every day, but I want you to come away with this understanding why algae need to sink or swim and why you need to keep it mixed. Okay, so at any boundary in an aquatic environment, this is inside the cell underneath. This is the cell membrane. And as water moves along the surface, there's a boundary layer where friction between the liquid phase and the solid phase slows down the flow rate. So the amount of diffusion here is relatively fast. Diffusion from here basically doesn't exist. And so this boundary layer and the friction is described by something called a Reynolds number, which those of you who do plumbing are probably familiar with. It's how much drag there is from friction in a pipe or something like that. It's something you have to pay attention to when you engineer a system. So you have to pay attention to this actually with algae too, so they can get at resources outside of that boundary layer where friction prevents diffusion. Okay, it's like a little insulating layer. And the way microalgae have solved this problem is if you introduce a bump in somewhere in the boundary layer here, you break up and make this an unsteady boundary. Instead of there being a very even decrease in the flow, you mix this boundary layer up with a bump, and thereby mixing some of the previously unavailable water and the resources in this water closer to the membrane so that the membrane can access those things. And so this is the explanation. Somebody said it like the shapes. This is why algae have such interesting shapes. And there's been a lot of conjecture about this over the years, that maybe it helps them float, maybe, you know, maybe so some of that stuff is true also. But a lot of the complexity on the outside of algal cells, these little processes and a dinoflagellate, these two are dinos, why would a colony be wavy instead of straight? I mean, that's extra complexity, and biology, that costs something. And then diatom frustrules are kind of famous for having all these little bumps and ridges and points and so on. Why are they so interestingly shaped? And the shape is now the best theory that science has is that all breaks up the boundary layer between the water outside and the membrane of the cell or the surface of the cell, where these enzymes are that are there to grab onto the resources, the chemical resources that the cell needs. And so, you know, that, I have another, no, I don't. So I'll, let me go back and mention, you know, the other aspect of this is algal cultures are referred to by engineers as a stirred bioreactor. And if you don't physically mix an algal culture, you actually impede the movement of water relative to the cell surfaces. And so we mix algal cultures usually with bubbles. And there's a reason to not mix algal cultures with bubbles, and that is the bubbles encourage bacteria. And that's kind of a complicated story of some, that's a good one for beer also. But our biggest algal cultures now, we had 20,000 liter tanks in a greenhouse. We're mixing them with a foil. And that was much more effective. We had sustainable cultures harvested semi-continuously here. In a 20,000 liter tank open to the atmosphere, so the contaminant, the most common vector bringing contaminants into our big cultures what were bird turds, birds flying in and out of the greenhouse. We solved most of that by putting a finer screen up and keeping the birds out of the greenhouse. So there's practical stuff like that. But anyway, you know, you got to mix in algal culture. You know, they will try. The diatoms will sink to break that boundary, to provide that flow between the surface, and the flagellates that swim are not doing that to get from one place to another. They're there to escape the envelope of water from which they have already accessed all the chemicals. So that's the reason of motion, and why it's important in algal cultures. So again, it's something I hope you know, when you see a poorly mixed culture is failing, you know, think about that. Is that the problem? Do I need to mix this a little bit better to you know, give the cells access to the resources they need in the water?